Good afternoon. My name is Francesca Dominici, and together with David Parks, I'm one of the two faculty co-directors of the Harvard Data Science Initiative. So I am delighted to see many of you today for our very first Harvard Data Science Initiative 5th anniversary panel on the topic of personalized health. The panel is a series of specialized events we are hosting this year to mark the Harvard Data Science Initiative 5th anniversary. Throughout this academic year, we will convene seminars, tutorials, workshops, and panels, some in person and some online, that showcase the work of leading computer scientists, statisticians, and domain experts from law, business, public policy, education, medicine, public health, and many other academic disciplines. We are absolutely delighted that Zach Kohane and John Quackenbush have agreed to moderate today's discussion, and we are grateful to our panelists, Preeti Hedges, Mark Namchuk, and Jessica Tannenbaum for joining us. Zach Kohane is a Marion Nelson Professor of Biomedical Informatics at the Harvard Medical School, Chair of the Department of Biomedical Informatics, and a Professor of Medicine at Brigham Women's Hospital. John Quackenbush is Professor of Computational Biology and Bioinformatics at the Harvard T.H. School of Public Health, Chair of the Department of Biostatistics, a Professor at the Channing Division of Network Medicine, and Professor at the Dana-Farber Cancer Institute. Both Zach and John are members of the Harvard Data Science Initiative Steering Committee, and it is my great pleasure to hand over to them now. John and Zach, now is your turn. I'm disappearing.
Wow, I was so enthusiastic and so grateful for your leadership, Francesca and David. I just could not wait to state my gratitude. So we're in for a treat today. I'm going to make two brief introductions, but then when it comes to the actual speakers, they'll reintroduce themselves formally. I will introduce Mark and Jesse and Professor Quackenbush can introduce.
John is fine.
Okay, fine. Okay, so we're really fortunate to have Mark Namchuk, who leads our efforts at the medical school in essentially bringing a therapeutics focus therapeutics and translational focus to what we do at the medical school, and has led several innovative efforts. He's also a veteran of the pharmaceutical industry, and what I've really enjoyed in working with Mark is his pleasant skepticism about how data science might play out with regard to development of individualized therapies. So it's not a gut reaction, it's just a thoughtful reaction that he has based on having been in industry and now academia. And so I'm still trying to have a gentle discussion with him about that, but I think that's very helpful. And Jesse Tannenbaum is a researcher at Duke who is on extremely prolonged leave running health information for North Carolina. And as such, it goes from one strangely Medicean environment of academia to an equally Medicean environment of state government. So, John, why don't you continue?
All right, well, thank you, Zach. It's really a pleasure to co-host this panel discussion with you today. Wonderful to have Mark and Jesse join us. Our third panelist is Dr. Preeti Hegde. Preeti has had a really illustrious career working in a variety of different settings. She began her postdoctoral life at GlaxoSmithKline, spent some time at Genentech, really developing at both those organizations, data-driven approaches to drug discovery and drug development, but recently joined Foundation Medicine as Chief Scientific Officer, really with the goal of trying to take these large-scale data sets and use them directly to help personalize care and direct care for individuals. So it's really wonderful with these three panelists to see the breadth of expertise that's represented. And so what I'm going to do as a first question to each of you is just ask you, what are your own perspectives on what precision medicine is? And what do you think the key challenges are in precision medicine? So I'll start with you, Mark. How would you define precision medicine?
Hi, John. It's nice to be here. Zach, thank you for the introduction. So nice to be the pleasant, the little bug in Zach's ear, I guess. So I think that when we think about precision medicine, I think the ultimate goal is, can you make an appropriate treatment decision for an individual? And can you coalesce a lot of science into making sure that decision is based on the most rigorous analysis possible? That's like our big long-term goal. I think that there's a ton of promise in it, and there have definitely been some successes. Maybe to just sort of go back to the introduction. So I spent 23 years in pharma, 17 of which I was at Vertex. So in part, working on genomic medicine and monogenetic illness. I think the challenge, going to the second part of your question, John, is illnesses that don't necessarily fit into that bucket. And how do we categorize them in a way that we are being more effective in being able to prescribe those therapies? I think the other part, and I think in talking to Zach about joining the panel, I think it's also taking a look at, where do you have the data in appropriate quantity and quality to make the predictions that would be most useful in, say, the drug discovery process, or ultimately in patient selection? And I think that while there's been innumerable advances in the computational side of it, I would arguably say, outside of a few pockets within genomics and being able to do large profiling studies, our ability to really think about pharmacologic action. How does a drug interact with the system? How do we appreciate the differences of dose? How do we appreciate the differences in selectivity as you modulate dose? To go back to what we're predicting, remains a place where our data sets are not particularly good, and I think haunts our ability to make more accurate predictions on a person-to-person basis. So I guess as an initial foray into the polite skepticism that Zach brought up, as a pharmacologist by training, I think that our work is not done in understanding how basic biochemistry interacts with these biological systems, while we've definitely made progress in understanding the underlying genetics of those systems.
I'm muted. Thank you so much, Mark. Jesse, do you want to share your perspective?
Sure. So I take a broader perspective, I think, on, and I've seen like tweets. I don't know if we're doing precision medicine or personalized medicine or both, but I've talked about these and say, I think of it as P-star medicine, whatever the latest moniker is that we've decided to call it this year or this decade. Fundamentally, there may be different emphases on personalized, precision, genomics, stratified, individualized, P4, whatever it is. All of these are really about using more and different types of information to provide the right intervention for the right person at the right time. I'm talking about people, not necessarily patients, because some of it even comes down to before you're a patient, before you fall ill, preventive measures. I think it's important to emphasize it's not just about the genomics. I think that all of us loved that shot of Obama next to the DNA strand announcing the Precision Medicine Initiative. But the problem with that is that it set us up for people to say, it's never about the DNA, just the DNA. Of course, it's not just the DNA. It's about so many other things. As Professor Kohane mentioned, I am affiliated with Duke and did research there, especially in precision medicine and mental health. But now I'm working in the public health space. I'm Chief Data Officer for North Carolina's Department of Health and Human Services. And so there's a huge emphasis in our department on social determinants of health. And so it's way beyond just the genomics and the physical mental health aspects. the economics, the transportation limitations, the food scarcity, all of these different challenges really impact a person's health. I think I will stop there. You're muted again.
I'm trying to be respectful and I keep on making my common mistake. So Prithvi, do you want to give us your perspective on precision medicine?
Sure. Before I do that, I do have the exclusive title of being John Quackenbush's first postdoctoral fellow way back in 2000.
And you survived. Congratulations, you survived. Amazing.
And thrived.
When we were working on the Human Genome Project way back when in 2000, we didn't realize the impact that it would have 20 years hence, where if you look at cancer, cancer is the disease of the genome. And today in 2021, practically every disease stems from an understanding and distinction of the heterogeneity of those diseases and the genomic associations with that heterogeneity. Having the ability to provide therapeutics that are tailored to address that heterogeneity really is how we view personalized medicine in both with drug development, but also in the context of diagnostic development. Yes, the availability of data, the availability of technology to sequence at scale and analyze data at scale has really revolutionized what we can do today with respect to new target discovery, with respect to how we develop clinical trials. In the past, let's take EGFR inhibitors. In the past, you needed thousands of patients in phase 3 trials to determine if an EGFR inhibitor had activity or not, because we didn't quite understand the association of mutations in EGFR and outcomes. Today, you can run trials in less than 2 years and get to an outcome that tells you if that therapy is effective or not. So great improvement in the way we design clinical trials. I think the challenge that we have is really on the public health space and the intersection between reimbursement, insurance, and making both diagnostics and therapeutics available to the right patients at the right time. And there's a lot we can do. And I'm excited to be a part of this dialogue here because you do have a public health school at Harvard. And I can't emphasize the importance of public health and economics to really shape personalized medicine and make it stick and land in the community.
So thanks, Prithvi. And thanks all of you for the introductions. In listening to the three of you, I see sort of two really interesting threads, right? One is genetics and heterogeneity. And in cancer and oncology, where Prithvi's done a lot of work, if you think about it, we often think about precision medicine as being single mutations, single genes that lead to our decisions for how to treat and even how we think about the disease developing and progressing. And I think one of the things we recognize is those single genes don't act on their own, that they're part of more complex systems. But it also touches on what Jesse's suggesting, that there is this public health component and the social and other determinants of health that do interact with these systems. So, the question I would throw out for all three of you, I guess, to start sort of a broader discussion is, how do you see addressing first the challenge of genetic heterogeneity, and then maybe we can come back later to the broader question of heterogeneity. And feel free to jump in.
So the genetic heterogeneity, something that struck me being in that space is just the sheer magnitude of numbers needed of people, of genomes to be able to get at the numbers. As we have the more variables, the curse of dimensionality, and the expense of clinical research and the labor-intensive recruiting, et cetera. And so I think the learning health system is going to need to play a major role in this, of just making it scalable, getting the ability to follow people over time, the phenotypic information. Hopefully we'll see more and more genomic genotyping done just on a regular basis without having to have special, the Baby-Seq project and all of that. And as we do, we'll have more data out there for everyone to mine. with ethically responsible, secure, private ways.
So you're talking about generating a lot of data, right? But genes don't act in isolation. So, Prithvi, how are you dealing with that? The fact that multiple genes can contribute to the phenotype and response to therapy?
This is a great question, and I think we're at a cusp, John, where we can really leverage genetic heterogeneity, especially with new tools like polygenic risk scores, where you're no longer looking at one gene and the impact of, effect size of one gene, but rather a cluster of genes that are associated with phenotype. One of the things that we've been there's two data sets, two types of data sets that one can look at to really inform the association between genetics and phenotype or disease. One is real world data, and I'll give you an example of the work we've done with organizations like Flatiron Health that have done a tremendous job of really emphasizing the quality of data that's collected from EMRs, from EHR systems, where you have, access not just to the treatment that patients are on, but outcomes to those treatments, toxicity profiles associated with those treatments, whole genome sequencing data available, various lab reports available, all of that together in the real world setting. Now, in order to analyze real world data, we still are at an, I would say at, in our infancy, really, in analyzing real world data, just because there is a lot of gaps that currently exist in our ability to fully leverage real world data. But on the second hand, you have well-controlled clinical trials that have, control arms, that have very well annotated data sets, large numbers of patients that many of our regulators have access to, the FDA, the EMA, the Canadian health systems. Having access to those data, if those data were made publicly available, I think it would vastly improve our ability to do genetic associations at scale. As Jesse mentioned, we need data at scale in order to address these effect sizes, which tend to be really small with genetic associations. But having access to those data is really helpful. And so, for example, at Foundation Medicine, we now have access to over 500,000 patients worth of cancer genomic data. And I'm talking about tumor genetics here, but that allows us to identify very rare frequency events, 1% of non-small cell lung cancer, or 1% of all cancers. That now allows us to identify new targets and identify patients that would match those targets. So not only do we do target identification, but we have the ability to match patients for clinical trials that would help in the drug development of those therapies as well. But yes, having availability for data at scale is really key here.
We can't hear you, Zach. Were you saying something?
I was just waiting for Mark.
Oh, sorry. Okay. I don't have too much to add, and I'm certainly not a geneticist on the scale of Preeti and Jesse. So I'm going to maybe take a step back and say that I think that part of the issue that we're all wrestling with, and I think, and this is something that I think as we get more and more data, more and more genomic data that we're going to have to think through very carefully, is ultimately a lot of the genetic signatures are around predisposition to get a disease. They are not necessarily predispositions to be effectively treated for that disease. And so I think a lot of the things when we think about genetic heterogeneity, they're typically going into the context of what we're going to do interventionally in someone that's sick. And I think this is, as we go into things like environmental factors and being able to really think more holistically about personalized medicine, there's this sort of, I would say, there's a discipline where we're going to have to think about what are we doing and what are the genomic associations for treating an established disease and intervening, as opposed to if we were going to try and prevent something and we're looking for predisposition of said. So Jesse had mentioned serious mental illness. The last 5 years of my industrial career, I worked on depression and schizophrenia. And there's a great example where in depression, arguably very little came from a genomic standpoint to say, here's the one target you want to go after. It speaks to the environmental and other factors that underlie major depression. And then the other one, which really sort of affected my thinking in this regard, was the emerging genetics in schizophrenia around complement, for example, which may be more important and arguably interventionally important in an adolescent, but not necessarily when somebody has diagnosed frank schizophrenia. So I think the issue of genetic heterogeneity, when you put it into treatment and response to treatment, is different from the issue of genetic heterogeneity for designing disease risk or segregating disease. And I think sometimes we conflate them a little bit, because we have had some successes going after, I'll use cystic fibrosis as an example, where we identified the gene, we reversed the defect by a small molecule, we saw clinical benefit. In these more complex polygenic diseases, I think that association is a bit tougher. And so I think that's where genetic heterogeneity meets therapy can lead to a difficulty in interpreting the data. Again, I'm not a geneticist, so I can't speak to how much data and when. I would leave that to much more expert folks on the panel.
So I'm going to interrupt the flow a little bit, because as well, Jesse, you wanted to say something, Jesse, go for it.
Yeah, let me just riff quickly. I think it was John who said earlier that the genes don't exist by themselves in a vacuum. There's genetic heterogeneity in multiple genes. There's also the clinical heterogeneity on top of the genetics. So warfarin is seen, when I was in grad school and learning about pharmacogenomics, warfarin is like this poster child for success of, we figured out the genes and we figured out the clinical guidelines based on the genes. And then when Rob Califf made me get up at some ungodly hour to round with him to see the patients and how this really worked, and there was someone who was about to be prescribing warfarin, and Dr. Califf asked, did you think of doing the genetic test? Like, totally wasn't an issue. It was a very elderly person, had many other complicating factors. There were so many other things, and genetics just didn't even play a role. So there's precision medicine, but that was much more about her comorbidities, her age, her fragility, all of that. So just want to mention that in addition.
That's great, and a good reminder that if Rob's lucky, you won't have him to kick around anymore, because he's going to DC as the commish. So I'm going to probably stifle a bit the flow, because as I was listening to all of you speak, I was thinking, what an amazing set of insights and talents we have here. Mark is talking about the growing evidence that complement is involved during development in schizophrenia. Jesse, you're talking about integration of clinical data with the genomics. Pretty is talking about a polygenic approach. And I'm thinking to myself, although our friends, the bubble that we live in, can speak this language fluently, who are the people? And given the fact that I'm looking, we have a lot of students tuning in here, a lot of students. How do they avoid becoming like many people in who are getting life science PhDs or are in medical school, and actually get very little of this practical, whether it's precision drug treatment, whether it's the big EHR integration? Where is that going to come? Or rather than putting it on the system, given the fact that we have these live students here, how do they best acquire the skills to do what you guys do and to be able to talk fluently as you have about these problems? Jesse, want to take a whack at it?
Sure. Yeah, as you mentioned, Rob will be gone, so he won't be able to boss people around that way. One thing, this was a point of contention among the students in my graduate program at Stanford, was we would come in and say, I'm a genomics person. That was actually what I said. I'm a genomics person. Why are you making me learn about EHRs? Why are you making me learn about all these other things, clinical decision support? And go to the talks that aren't on what you're studying. Go to the symposia, go to the everything, and just really expose yourself to the full range of things. It will serve you well in the future, because there's so many of these sort of data science concepts that are common across the genomics, the molecular, the imaging at tissue level, the clinical at person level, and the public health at the population level. So don't have a focused mind, stay open to other ideas.
I'll just say that it takes multiple disciplines to come together to make anything happen. And it's usually the most boring, the most mundane thing that becomes the most important thing that you need to do in order to really realize what your vision is for that activity. And that's when, go ahead.
Could you give an example of that? I love it. I think you're right. I think absolutely think you're right, but can you give an example for our students?
I'll give you an example, Zach. We had this vision of, when I was at Genentech, we should collect data from every patient on our clinical trials, and that data will transform drug discovery and so on. You won't believe it took us over 5 years just to get systems in place where data sets can be put in place in a manner that allows us to analyze data, that we need to have cloud compute architecture, that we need to have data access policies. These things are things that may be kind of boring for a scientist, but critically important for us to get to a place where we can actually start to analyze the data and make something out of it. That's just with one example, but I've noticed that with everything in life. I'm at CSO at Foundation Medicine, and I realized I can innovate till the cows come home, but if the insurance providers don't see value in what we're doing, and if we can't demonstrate value for patients, there is no point in innovating. So it always comes down to the practical, boring, mundane things that drive true innovation.
Maybe I'll say something that bridges in between the two, because I completely agree with both points. I think the most eye-opening thing for me, so I love to use this phrase, that biology will teach you great humility no matter how hard you try. So this idea that we've got it all figured out is a temporal thing. And the reality is that our ability to ask the questions we would love to in more complex systems, like being in the clinic, is often limited by the practicality of running a clinical trial and ethics and all the other important things that you have to make sure to do things properly. So I would say one of the most eye-opening experiences of my life was being in a pharmaceutical company and thinking about design of a phase 2 trial for a cancer therapy, where you've done all of this wonderful, pristine biology, and then you need to figure out how you're going to ask that question on top of standard of care. Which patient populations can you get to? Most patients are not going to sign up for serial biopsies. So I think there's this, it's kind of what Preeti was saying, there's this practical world that you're going to run into. And so going back to the students, talk to a trialist. If you're really thinking about being in a career in cancer genomics, go out of your way to go and talk to someone who's had to design, particularly in industry, a phase 2A/B trial, just so that you know what you're signing up for and how hard it can be to ask that very precise question. And I think the other thing, which is really building on, I think, what Preeti had said, I think that my most aha moments come from an incredibly bright but naive person that knows nothing about your field, that walks in and sees something like, we're also group speaked. Oh yeah, it's got to be this. Oh yeah, this is super important. And someone who's not in that field at all walks in and says, well, what about that? And you go, oh, right. So, again, from a student perspective, it's exposing yourself both in your individual research, but also in the classes you attend, in the seminars you go see something that's just challenges your own common belief, just to make sure that that belief isn't group speak as much as you haven't missed some flaw in it. And again, I can tell you from running a research group in industry, those were the most amazing moments when we'd invite someone and people are looking around like, why is that person there? And then they would say something like, oh, well, you should be looking at this. And everyone would have this dumbfounded look of, oh yeah, we should have looked at that a year ago. Great. So I guess that would be my add to the previous comments.
Well, I love that perspective, Mark, that, two things. One, dumb ideas are often the best ideas. And it's often somebody coming in with a new perspective that really simplifies the things you're looking at, that you get so deep into the weeds, you forget where you're going. But I think the other thing you said that I found really intriguing is this idea that biology is humbling. And, I've seen that all the time. I mean, I'm somebody who loves data, loves new methods, new approaches. And, to commit heresy here with Francesca and David on the data science panel, right? I mean, one of the things everybody gets so excited about is machine learning. And we can take these massive data sets and we can just throw them into machine learning, and somehow the linear algebra magic is going to spit out truth. And so I have my own perspectives on that. But, I'd love to hear from each of you what you think the best road is forward, given the fact that biology often has a lot of tricks up its sleeve. So how do we approach this? Is it by trying to do a better job of phenotyping, trying to build better analytical methods, or any and all of the above?
Not the analytical methods. That's what I was. So not that it's not important to have good analytical methods, but that's not the bottleneck right now. The bottleneck is, as someone just mentioned before we got on, asking the right question. I show a slide in a number of talks I've given where I show like the deep learning network with many levels, and say this is what academics are worried about, when really what they need is this like select statement with appropriate group by and summarize. It's a little more complicated in genomics and biology, but that aspect, it's really the actionability. What do we do with this? What can we change about what we do day to day if we were to know this? It's the implementation. It's how do you build that into a workflow that's extremely entrenched with clinicians and providers and genetic counselors and everyone doing what they do. Make sure to get it in front of them in a way that's going to be digestible to them in the busy day they have. And then there was one other point I wanted to make. Oh, and then ultimately, so much of it comes down to eat right and exercise. And people can know that, and that doesn't mean they're going to do those things. So figuring out the precision way to get people motivated to do it. I don't know if we've cracked that nut yet.
Prithvi?
I read the article, John, the one that someone interviewed you, I can't recall, on machine learning and the pros and cons of machine learning. And you said it really well. The most important thing in this day and age of using computational tools is having a training set and a valid independent validation set. Having independent validation is so critically important for us to know if what we've identified is a true signal or an artifact of that training data set. In this world, especially when you're going after black box algorithms where you don't know what your hypothesis is, you don't know, if the biology even exists in that disease that you're investigating, having a training and an independent validation set is critically important. And I wonder if, frankly, even with publications, if we should accept publications that have just training data sets without independent validation.
So I have a question that will probably make people regret that they ever asked me to moderate this panel. So in COVID, we've all been looking for treatments and also for cancer. And there seems to be evidence that the experience might actually be different by your continent of origin, where if your parents came from Asia, or your parents came from Africa, or you're a child of slaves, or your parents came from Europe. Given the fact that we are much more acutely aware of these issues now than even, let's say, 2 years ago, how concretely do you think we are factoring that into our study design, into our just better, processes to make sure that things that we develop work well for different parties? And Jesse, this also applies to you because SDOH might actually get expressed differently in different ethnicities. And so on the one hand, you want to be focused on the individual. That's the right thing. And we've shown that, well, others have shown that things like that a data-driven approach can actually substitute for a race label in, for example, estimating glomerular filtration rate. But that's not so clear for other things. And so how, I'm not looking for solutions because it's a complex problem, but how are you beginning to think about doing that and serving communities or target populations? Priti, looks like you want to say something.
Yeah, this is a really important question, Zach, and there's a lot of work going on now to ensure that we have diversity in clinical trials. We have ethnic diversity in clinical trials. I'll just give you an example. Predominantly what we see today, unfortunately, is the Caucasian population, right? Our clinical trials are run in the United States or in Western Europe. When we start utilizing tools like whole genome sequencing to say, hey, are there signals associated with either efficacy or toxicity? We've come to realize that most of what we're seeing is unfortunately needs that label of only in Caucasian populations, because we don't have Asians, we don't have, Black communities represented. Most of clinical trials take place in AMCs, in affluent communities. One of the things that's happening now, at least in the community setting, like, organizations like One Oncology, is that they have a concerted effort to ensure that patients in the Black communities have access to clinical trials. We within companies, whether it's diagnostics or pharmaceuticals, are also doing a lot now to start to ensure that we have that diversity represented in our data sets as well. But you're absolutely right. So critically important in this day and age to ensure that either we have representation or that we have these things in our label saying that, well, this is only for a specific population.
Mark.
Yeah, I think that I'll just add two things to that. I think that if we take a big step back, this is why when one thinks about worldwide approvals, that you often see different trials run in different countries. So I think there's been, I think we have to take a step back and remember it's been recognized for a long time that medicines will work differently in different populations. It's a separate issue from inequity in recruitment. But I think it's been long acknowledged that we have to make sure that we're doing a good job. If you're, for example, getting an approval in Japan and you have an inadequately studied Japanese, you will have to run a trial in Japan to get that drug appropriately labeled. I think going back to your COVID question, Zach, and to kind of a panel you and I were on maybe a year and a half ago, I think that there's sort of two parts to this. I think that if we think about COVID therapeutics, and let's divide them in half. Let's think about therapeutics that treat the inflammatory components of the disease, and then say targeted therapeutics that go directly after the virus and just kill the virus. I think the data are really strong that ethnic background, other factors will diversify the response in the anti-inflammatory part of the disease. And so part of it's also just being rigorous scientifically. It links back to labeling, but understanding that patients' immune systems are going to be different by background, exposure, et cetera. And in order to appropriately label, you have to appropriately study. Part of our issue in COVID was there were so few large enough, well-controlled trials that were being done, particularly early on with repurposed agents, that I would arguably say at all levels, we hadn't adequately powered to make conclusions. I think that what you're seeing now with the emergence of, for example, Pfizer's protease inhibitor or molnupiravir, which were designed directly to go after the virus, that you'll see less variability by patient with that mechanism. But to the point that Preeti was making, you got to prove it. So I think part of what we also need to recognize is that different disease areas with different interventions will have different demands to ensure that you really appreciate all of the biology that is going into whether a treatment will work or not. One of the things about direct antivirals is that they have a relatively simple pharmacologic thesis. You are shutting down the virus's ability to replicate, and you're then looking at whether that has patient benefit. And so I think that's where it's a little different from cancer, where cancer is always integrated into patient's background genetically, ethnically, et cetera. So infectious disease is a little different that way. And I think we have to think about it, at least in those two boxes. What Preeti's saying is absolutely critical. If it's on label, it has to have been studied, and we have to do everything we can to make sure we've looked.
Well, sort of related to
Jesse.
that, I'm sorry, Jesse, but, sort of
Jesse.
related to that is that one of the least studied problems in health and medicine is just sex differences that we know in diseases like colon cancer and COPD. There are profound differences in risk in natural history of disease and even response to therapy between males and females. And yet it's something which often isn't considered in a lot of studies. So I'm sorry, Jesse, I interrupted something you were going to say.
No, I was going to say something related to this discussion, just to put in a plug for the importance of explainability as we get into machine learning, AI, to figure these things out. The importance of being able to say why we got to give an answer and not have it be black box. Also, a quick shout out to my friend Cynthia Rudin, who recently won the second year of the, AI for the Benefit of Society award, $1 million award for using AI in this way. And she works on explainability and the importance of that. And I think less in genetics, because there you're really getting to something more fundamental than race and ethnicity. But in other areas, saying why you came to a conclusion is going to be really important to make sure that we're not being biased by the data sets that we're training on, or something that wasn't what we meant to be looking at, that was based on bias in the way we already practice medicine and health.
Yeah, so one of the things you mentioned AI systems, and one of the things I'm really interested in is the need for greater openness in science. And I think one of the challenges we've seen as a community is that as data sets get larger and more complex, the analytical methods we use to try to tease out what's in those data sets has to sort of grow in complexity. And as we want to take these from discovery tools and investigational tools and move them into the clinic, that one of the things where a lot of these methods kind of fall down, or one of the places they fall down, is a failure to make not only the data available, but the codes and the models that are used. So, as you think about treating patients and moving these into the clinic, how important is it that we really understand what the methods are doing when they make predictions, since you mentioned causality, Jess?
Very, very important. All the more so when it's these huge data sets. I mean, in grad school, I lived in fear of an off by one error. And if you don't have access to the data and the code, you can't prove that something like that didn't happen. So I think it's really important as we get these large data sets to get that out there and have people have access to what you've done and be able to reproduce it.
Journals are now asking for authors to make data sets publicly available. And I think that's a great first step, John, in enabling public access to your data sets and enabling others to mine and independently validate your biology, the biology you've identified.
But what about the tools and methods, right? So if you have a machine learning predictor, or even a statistical predictor of some outcome, right? The data is okay, but how do you prove that your conclusion is right?
How do you prove that your conclusion is right? That's like a seriously deep, fundamental scientific thing. Yeah, I think the move toward open science and reproducibility and sharing that is key to that.
Yeah, I think maybe another point, and again, if we're thinking about this as a clinical diagnostic, John, I think it does go back to the rigor with which you validated that across a broad enough population to say that what you're using it for is appropriate. So there's a definite thing about transparency and reproducibility from a scientific perspective, but our other audience is really how predictive is this from a treatment perspective? How well does an individual patient feel? What's their confidence in using it? And sometimes that's less about sort of the underlying 12 lines of code and all the other pieces that this audience would well appreciate. It's more about how well it's been studied and validated clinically, and being transparent about how predictive it is. I mean, this is something that I think in the cancer field is an interesting example where biomarkers are critical, absolutely critical. The positive prognostic value of a treatment outcome for most biomarkers in cancer is still not at the point where it's a light switch. And so I do think there's sometimes when they don't work, then you create a certain pessimism, and how to communicate statistics well to a broad audience is something I think that's haunting the scientific community, well exemplified by what's going on in COVID in the media right now. I think it's just a really complicated issue.
Well, some of that comes back to what you were talking about earlier, right? With a well-designed trial, that if you want your results to generalize, you've got to really design the study to make sure you're ticking off all the boxes, you're not seeing hidden effects.
I'll just add one thing to that, John. With well-designed trials, your inclusion-exclusion criteria can be so narrow that really you're only addressing a very small patient population compared to the general patient population. And I do wonder if, in addition to well-designed controlled trials, you do need to have some level of real-world evidence to say the patient that walks through my door will respond equivalently to what a clinical trial patient would have responded. And there's still a lot of work we've got to do to address that.
Could I jump on that train? Because I feel very passionately that Preeti's correct, but it's also, just to almost expand the book a little bit more, there's the narrow path you have to walk to get something approved, which is often a driver of those criteria, both from a cost and pragmatism standpoint, because you have to ask a specific question that you agree to with a regulatory agency that if I called my shot and I get this answer, this is an approvable endpoint. And incorporating real world data into that in a rigorous and statistical way, I don't know that we've got a great framework for doing it. It's anecdotally added. It was part of the expansion of the PDUFA guideline. But even from sort of a rigorous analysis standpoint, it's a very hard thing to incorporate into the approval process. But I think it's a missing link, and it's not something I don't think we as a field know how to do it well.
I fully agree, and someone that works with real world data all the time. But this is actually, I think, for the students particularly, just an incredibly important point. They don't even know about the pressures to make it narrow or wider. So you want to make it generalizable, but the more patients, more heterogeneity in your patients, the stronger your signal is going to have to be in order to beat the standard deviation that it's actually in effect. And so doing those trade-offs are hard, and finding the right data sets that don't have weird ascertainment bias is pretty tricky. In fact, as part of the Roche acquisition, Flatiron, that was, I think, one of the hopes was that this would help define classes of patients. We'll see how it goes.
I think it will also help now, Jesse, if you have influence with Rob, Caleb, and others, is we need educated folks on the regulatory side of the business as well to say, what does good need to look like? So it's not just about, academia and industry, but regulators need to have that vision and that education to say, this is what good needs to look like, and then provide guidances to all of us to say, what does that data package need to look like? Can I ask a question of John and Zach?
Go right ahead.
One of the great advantages of large scale data, I think, is what the UK Biobank has shown, right? I mean, this is years of data collected at a state level with, outcomes data, lab information, whatnot, and all of us have the ability to mine it, which is phenomenal. Why is it that the United States hasn't been able to do something like that? I mean, we do have the NCI, we do have the NIH, we have prestigious organizations like Harvard. How do we develop our own US biobank?
So let me answer that, because initially, I was very skeptical that the UK Biobank was going to be successful. because there were a group out of Sanger Institute who understood the genomics, and the National Health Service who did the clinical. They didn't even know how to talk together. So there were literally years where it was like, biggest wasted meetings ever. But somehow they've developed this amazing resource, unprecedented resource, which is truly transforming thousands of labs in their ability to do research. And I thought we had a edge with the precision medicine initiative that Obama announced. I was actually in the White House as an onlooker at that point, but we lost our edge. And I think it has to do with, I think there's a greater sense of ownership and citizenship in the community, the public in the UK. They're proud of the National Health Service. And so when people come up and say, we're going to do this, the doctors feel part of this healthcare system and not part of these balkanized healthcare systems, which frankly, whether they're for-profit or non-profit, are still trying to make money. And doing research is not exactly front and center. And they each do business a little bit differently. And so having, with the lack of a public trust in a broad National Health Service, with a very balkanized, more opportunistic kind of healthcare system, you can't get the basically the agreements that you need to get the broad consents and the broad data deliveries that working at scale would have. John, you want to take a whack at that?
Yeah, I think Zach touched on a lot of really important things. A phrase I often use to describe healthcare in the US is balkanized, right? It's not just fragmented. They're groups that actively don't want to work with each other. And one of the big advantages I see in the UK Biobank is, even though there's a little bit of variability, there's something greater approaching a standard of care that's used for everyone in the system. There's something closer to a universal health record. And I think one of the biggest challenges we have in the US is, and I alluded to this earlier, it's sort of defining phenotype, right? We all talk about health and disease, but in each one of those groups, there's a broad diversity of what we mean by health and disease. I mean, not tumor is easy, but, do you have, breast cancer? What kind of breast cancer? What are all the factors that affect it? So I think that's easier in a system like the UK. In the US, we do have some attempts to try to build that. All of Us is a great example. TopMed is a great example. The Million Veterans Project is a great example. But, like Zach said, I don't think any of them have overcome the barriers that the UK Biobank has. I hope we can do that, but, I'm not sure we're there. Maybe some of you have better ideas for how to do it. Go ahead, Zach.
Yeah, so I think that's extremely well said. And we see the same difference that Preeti talked about in the approach. Let's agree, I hope Mark is going to agree, that the single most important drug that was developed during the first year of, that was delivered during the first year of COVID was not from a pharma, it was from pharma long ago, and it was dexamethasone. And dexamethasone was a result of the fact that the UK could get its act together across multiple hospitals and run a fast, stripped down RCT. And we could not get our act together to do that in the United States. It's pretty sad, actually. And it also speaks exactly to the lack of alignment across institutions and the ability to people say, I don't care what our revenue model is. Here is how we're going to bring together the data. And they did it. And it's actually this nice article in Science from a year ago about that, what the process was. And they had to basically strip down IRB issues, and simplify data collection, but they did it. And because they were all working together in this very voluntary spirit, it was an enormous boon for patients. And it's sad that we were not able to do anything similar like that in our country.
Yeah, just maybe add one comment to that. I think that the other part goes back to linking back to something that I think both John and Zach said, it's a national health system. So it was in the best interest of the country to provide an evidentiary basis for the use of dexamethasone. Right, because it's a generic medication. It's not going to be done by a company. So if you think about medication in that circumstance as an extension of public health, I think that's a big chunk of it. And I do think that that's a place where the US needs to take a little bit of a look at what's been done at Europe, even in particular, the use of medications that are now either generic and no longer branded, where a company's not necessarily going to put the investment in to get that evidentiary basis. And I think it's fascinating because, for example, there was no need for approval for use of Dex in the US, but in Europe, they actually did a label change, right? So it just kind of goes to the differences in the system. And I think one of the things that I'm fascinated by, I don't want to chase a squirrel down in the field here, but it's this: how do we, in something like the pandemic we went through, create ways to get appropriate evidence for medical decisions when all heck is breaking loose? And this goes back to your point, Zach. The UK did a much better job of pulling things together to start creating that evidence more quickly. And I think it's a model we have to look very seriously at.
Well, and one of the things about trials, right, is a lot of trials fail because they can't recruit enough patients. And part of the advantage of having a really well integrated system is you can do some of that. And I think that's a big advantage of a national health system.
So I see two questions.
Yeah, I was going to tackle one of them, Zach.
Go for it.
So one of the questions I got interested in, somebody asked about behaviors, and you just mentioned public health, Mark. And Jesse, early on when you introduced yourself, you started talking about sort of the broader healthcare ecosystem, right, which includes public health, things from basic nutrition to exposures and behaviors. And one of the questions was about behaviors in chronic disease and how we start to, I think, both collect that kind of data, but also incorporate it in our decision making. So anybody interested in tackling how we might want to do that? And I look at you, Jesse, simply because you're the one who raised things like nutrition.
So, I don't want to conflate behaviors with certain social SDOH. Actually, SDOH has gotten sort of a bad rep around here because there's some objection to this idea of social determinants. Like, well, it's sorry, you're poor, so you're going to be unhealthy. It's not deterministic. It should be like social factors in health. So I think the question was about behaviors. How do we target that? I honestly I don't know the answer to that. I think it gets back to what we were saying earlier, is I can tell you based on your genome what you should do: eat right and exercise. Zach is illustrating the importance of this for us. Thank you. I remember it was Steve Quake who had his genome done, and then they found some heart risk allele thing, and he was supposed to be taking some drug, and he stopped. He did where's the gene for compliant with what doctors tell you to do? I don't think we found that one yet. So I'm speaking up to say I don't think there is a good answer.
I think so.
Go ahead, Mark.
So I kind of going back to mental health, because Jesse had raised that too. And again, it's an area I'm super passionate about continuing my work on since I've come to HMS. But I think that behavior in mental health is not separate from biology. And so I think there are places where because the brain itself is adapting and learning, and so much of what you might see as a manifestation that's diagnosed as disease is how you adapt to a situation, all of the other people. So every medication that's used in psychiatry, I'm being a little hyperbolic, but I think this is close to true, works much better if you have counseling paired with it. And there's real physiology that backs that up. So I think an area where we have to stop thinking about behavior as this sort of separate variable, separate from medicine, and where it's a little easier to connect is the effects of environment, behavior, intervention, and neurobiology, and how we have to pair that with something that's pharmacologically intervening, as opposed to seeing them as two separate variables all the time. So I think that I'll give one other example is people are fascinated with the microbiome. I find the microbiome both fascinating and unbelievably frustrating because I have no idea what to do interventionally with the microbiome. But there's another example where habit, what you eat, behavior is actually altering potentially your immune response in potentially a measurable way. But we've not paired those things up. So I don't know if that's directly asking the question that was in the chat, but I think there are some disease areas where they're more closely associated than maybe we would admit.
Well, one of the really, oh, go ahead, Zach.
I think these are really great points, and I'm glad that we're focusing on mental health, because by the way, speaking now as I have a PhD in computer science, but I was trained as a pediatrician. There is an unbelievable disaster happening in pediatric mental health right now. And our local hospital is not accepting elective admissions, not because of COVID, not because of trauma, because our psych wards are overflowing. And this is, by the way, not just us, it's nationwide. But here's the problem. I totally agree with Mark and with Jesse, but the question is, the data come from two different places. I know how to measure LDLs, but it's harder to measure, activity level. People have tried, but there's been this disconnected community around wearables and the medical data, and they don't, and so they have companies that are trying to make themselves digital health companies somehow with your Fitbit or Apple Watch. I'm going to diagnose you, when in fact if we could figure out, and doctors don't want this data because that would overwhelm them, but if we could figure out how to join in a useful way the clinical data with these measures of life outside the hospital, that'd be great. But I don't know. Mark, have you seen something going on in that space that you admire or makes you hopeful?
No. I mean, I think it's also not a field I dive a ton into, Zach. You're much closer to it than I am. Maybe some of the other panelists have seen something that would be more kind of in that direction.
My colleague, go ahead, Jesse.
So I could speak for hours about this. I'm fairly passionate. Before I left to go to public health, mental health really was my focus. And before I knew I was going to public health, I agreed to co-edit a textbook on mental health informatics. So there's so many things that could be said here. mHealth is a huge opportunity. Natural language processing is a huge opportunity because so many things are, described in prose and not captured as a blood pressure number or something like that. It's also important to remember that as opposed to cardiology, where we kind of understand how the heart works and how to quantify that, there's just different epistemology of like understanding what is knowledge. There's different concepts. They're all self-reported and subjective as opposed to something we can quantify. So there's a huge number of challenges in that space, but totally agree with, I think, Mark, you were the one who started this conversation of like, it is critical to integrate these, and especially COVID has made it all worse, the mental health pandemic we're seeing. So yes, very passionate. Available for pre-order on Amazon.com.
Prithvi, you're going to say something.
No, I had the same point as Zach, that we have low-hanging fruits today with wearable devices, but whenever it comes to continuous variables, we kind of get lost. We just have no idea how to integrate continuous variable information.
So my colleague here at the School of Public Health, JP Onella, he's actually really embraced this idea of digital phenotyping, which I really like. And he started off with wearables. But then he recognized that, people take off their wearables, they don't charge them. But the one device they never forget is this. And it has all kinds of trackers. You have the ability to look at things like the complexity of text messages and the use of language. And so he's really been working toward trying to do better digital phenotyping in neuropsychiatric disease, right? If you're depressed, you sit at home and don't move. If you're happy, you go out and you do things. So even just motion tracking can provide a lot of information. But it comes back to, I think, one of the broader challenges we all have in doing biomedical research, which is defining the phenotype. So I think there are opportunities to move this forward. But, like Prithvi said, the real challenge is with this huge continuous data and how you make sense of it. One of the other things, go ahead, Zach.
No, I was going to change topics, but David Shaywitz. By the way, David Shaywitz is a great example of the strengths and weaknesses of behavioral change. So David is almost 2/3 of the man he used to be. And because he went with a for-profit company that gives you coaching and on a ketogenic diet, and he went with it at a passion, but it's very, very hard work. And I know many who have tried what David did and have failed. On the other hand, I can tell you there was a very exciting article in the New England Journal of these GLP-1 agonists. You give them to patients, they lose weight, and they continue to lose weight up to 60 months. It's crazy. And so the question is, do we have enough leverage to do behavior modification? And are there enough David Shaywitzes out there so we can succeed? Or should we just say, you know, humans are weak-willed individuals, and if we really want to combat the biggest epidemic in our lifetime, namely obesity, we're going to have to go pharmacologic. I'm being purposely controversial, and I wonder if anybody wants to take that on. Or if I could get David to speak, but unfortunately he's just asking questions. All right.
John,
go ahead, Jesse.
Where was the question in there? So David did amazing things. What are we discussing?
What I'm discussing
So
is even if you can identify mental health issues, A, they're squishy, and B, if the sort of behavioral characteristics are what we're trying to modify, that turns out to be very hard. We don't do it. Facebook knows a lot more than most of us know about how to modify behavior, but the healthcare system doesn't know how to modify behavior. Health insurance companies don't know how to modify behavior. They've tried all sorts of incentive programs for cigarette smoking, weight loss, and so we just don't know how to do that. And we failed at scale. And so that's my point. My point is, not only is it hard to quantify and get the right data on these mental health data, but if the point of attack is the behavior, and I completely agree with Mark that the data does suggest that combination pharmacotherapy and human therapy are as most effective, but that human therapy is expensive and equivocal results. That was the point I was trying to make. I mean, you have responsibilities, Jesse, as Chief Data Officer in North Carolina, in the sense that you have, I'm sure, outbreaks of mental health problems associated, for instance, with COVID. How are you helping, if you can talk about it publicly, identify those subgroups and make sure that perhaps they're being funneled to the right care provision?
So I wish I could describe phenomenal success we've had in that area. We know that there's a huge mental health crisis in the country, North Carolina not being any exception, and that COVID has made it worse. But I'm proud to say over the past 18, 20 months, we finally have a system where we're able to quantify the COVID issues. How many people have gotten vaccinations. One area we've made a particularly strong push is demographic race and ethnicity data. So we're finally pretty good at that. The vaccines, the cases, the testing, the medical surge, all of that. The mental health is a whole area where we don't have the infrastructure yet. So this is another thing coming into the role. We think about the sexy predictive analytics, machine learning, what can we do? And you get in and realize, oh my God, they're using faxes. Like that's where we're at. So the sort of data, IT, infrastructure, governance, quality, automation, those are the things we need to get into place first. And then ask me next year about what we've managed to do with mental health and public health.
It's the boring, mundane things first.
Absolutely. I literally just this morning so we were drawing on a whiteboard, and I was like, can I just say this is the part of my job I hate? Like, this is the boring, it was like logistics of how does someone request data. It's not sexy, it's not exciting, but it's extremely important. And it turns out to be a huge obstacle in many places.
People used to come to me and they say, oh, you like to build databases, don't you? And I say, no, it's about as much fun as getting a root canal. But it's one of those things you have to do if you want to chew on the data and do something with it. So, I feel all of your pain.
I really quickly, John, I think you make a really important point that from the behavioral aspect, the chronic diseases are key that way. And in terms of, I mean, we know in America there's an obesity epidemic, and that is that key area of like, we all know what we're supposed to do. That didn't prevent myself and others I know from putting on that weight during COVID. And so let's see what we can do now.
Yeah, so one of the questions that appeared in the question and answer session was about the human cell atlas, but I think about it more broadly as single cell data. And one of the things that's really interesting to me, I think a lot about biological complexity. And I can take that from individual genes acting in networks to sets of cells acting together in tissues to diversity of individuals and populations. And when I think about single cell data, one of the things that really strikes me is that the driver of innovation in health is new technology. So, I wonder, in your own work, number one, what you see as the value of data like single cell data, and then also what are the technologies you're excited about leading us to new types of information that can shed light on some of the issues we're talking about. And it might not be molecular, it might be just better data collection tools in the public domain.
I think the immediate real world application for single cell is in target discovery, John. I'll just give you an example. The whole space of CAR T cell therapies, for example, very exciting, cutting edge innovation in cell therapies. But the most important aspect of CAR Ts is identifying cancer antigens that these CAR Ts can more, specifically home to. Now, cancer antigen identification is a big challenge because we don't fully understand what are the different cells that these antigens are expressed on. And that's important for toxicity and off-target, off-disease toxicity, so to speak. This is where single cell sequencing is going to be really important, is how clean is the target that I'm going after, and are we going to get off-target toxicities if we were to develop this therapy? Second one is, I think Kai Wupferching just had a really nice example on CD161 that they identified through single cell sequencing and made a company around it, again, from a target discovery perspective, but it came from single cell sequencing data. So huge opportunity for target identification.
I think one thing, and maybe it's an adjacent technology, but something Zach and I have talked about before. I think that the recent revolution with things like AlphaFold and three-dimensional structure prediction from just de novo data, not quite ready for prime time in terms of ligated structures and those sorts of things from a drug discovery perspective, but to genuinely begin to better computationally predict the full pharmacology of a drug and be able to then appreciate how that affects biology. I think we're just scratching the surface of what that's going to be. I think that what haunts us, again, I think it's both from, I think about it from a treatment design and then from a patient selection perspective, is it's so difficult a priori to know everything a drug is doing, particularly for a small molecule. So we identified this gene, this gene, this gene, but we might be hitting 20 or 30 other things. And I don't think we understand it very well, and we haven't been able to incorporate it into the way that we think. But I think we're starting to see, and Zach knows, so this was one Zach nailed me on. I was completely pessimistic about AlphaFold until I saw 4 or 5 structural biologists that I trust play with it, and they were terrified. They thought this was going to put them out of business. It is an amazing advance, and as that gets more useful for design of ligands, I think that's something I'm pretty excited about. We've really never had the ability from a drug discovery perspective to fully enumerate selectivity, and let me be precise here, not to make it completely selective, but maybe to add good dirt, be able to tune systems as opposed to always have to go to the one lock, one key definition of disease. We've just never had tools to do that in any thoughtful way.
I fully agree, Mark. And talk about making algorithms publicly available and data sets publicly available. What a great example. This is as impactful as the Human Genome Sequencing Project is AlphaFold.
Well, and
John, that may be a fantastic, optimistic closing statement. What do you think?
well, you know, the great thing too is that your key and my key might be different. And I think that's where we get this intersection between genomics and structure and function.
Yeah. All right. So I'm going to ask my my lovely co-host, John. Can we bring our panel to close? I know there's some more questions, but I think we've actually had a great discussion. I think the energy is right, in my opinion.
I think it's a good time, but I think we should allow our panelists to make closing statements. Before I do, I know you gave thanks to Francesca and David for their work in organizing the Harvard Data Science Institute. But I also want to thank Elizabeth Landon Gray and Kevin Doyle, who did all the footwork to put this meeting together. And before I let them say have their final word, I really want to thank our 3 panelists today for an absolutely lovely and engaging conversation. And the dog. I have about 50 extra questions I want to ask. And I'd love for all of us to be together in a room with a glass of wine to be able to sit and chat. And I also want to thank everybody who joined and asked questions today. But let's have closing statements, and maybe we go in the reverse order of how we started. So Prithvi.
Well, thank you for this opportunity. This was really fun. I guess to the students, there's a lot you can do in this space with data, we just talked about a few things here and how you can combine behavioral data with genomic data. Would love to hear what you guys are up to in this space. I saw a few things in the chat Q&A as well with smartphone apps and so on. This is just the beginning. Of course, we've got a huge way to go with consents and ensuring that we have the ability to do what we want to do. But it's an exciting time to be in the sciences and in personalized medicine.
Jesse.
I second all of that. I'm always really excited to see when mental health and mental illness enter these discussions, because I'm just glad they're getting more recognition, stop the stigma, all of that. I think as we see more and more the biology underlying behavioral issues, it won't be so stigmatized, and it'll just be precision psychiatry. I second the get involved students. There is so much that needs to be done. There's so much that needs your skills, passions, talents, and energy. Reach out, ask questions, network, rely on your mentors. And there was a final thing. Oh, and my last, the last little thing. So I fell into public health really by accident, public health population informatics, especially through COVID. One thing that has become glaringly obvious is the disconnect, the unnecessary disconnect between public health and clinical care and the data that moves around those places. And so I think there's huge opportunity there. So I want to say academia is lovely, but it's also really invigorating to be outside of the ivory tower and having an impact. So don't forget your local, state, and regional government entities that may need your data skills.
And Mark.
So again, my thanks as well for the invitation. This was a lot of fun and very nice to meet the folks I hadn't met previously. I think that I'll make kind of a closing remark, both a request, but also a request to the students, but a more general request. I think one of the things that's haunting the informatics field as it becomes more and more dominant and bigger and bigger and bigger is just making sure that one of your first instincts is not that I'm going to be able to model something in the absence of input from my friendly biologist, but rather that my first instinct is to go find a friendly biologist to figure out how and what I'm modeling and to potentially give me a little bit of intuition about where I might want to go. I don't think our goal is to take the human element out of drug discovery. Our goal is to be able to harness these extraordinary advances in computation to make all of these things more efficient so that we can do a better job at impacting human health. And so we talk about siloing a lot, but I think it's also just, it's not just that we're in different buildings, it's also about remembering what the broader goal is outside of your individual discipline. So to the students, I guess my request would be, even if you're an extraordinary data scientist, you love the computational stuff, the mathematics, all those sorts of things, find folks as you're trying to put this particularly into the real world to be your co-pilot, and you'll both learn a ton. I've been so, my mind has really been opened up a lot by working with Zach and Robert, a person that Zach brought on board. I didn't know anything about that world, and I'm old. So being able to start that journey 35 years earlier would have been amazing. So I think that really just remembering the goal isn't just to have a computational algorithm that's going to predict everything. Is to plug that into the ability to move human health forward and find friends to do it, I think would be my closing comment.
You're muted, Zach.
Still muted, Zach.
This is the way I like hearing from Zach the best, when the sound is off.
Yeah, all right. I just saying, John, that was a wonderful, Mark just did a beautiful ending. I think it was the right message for the right audience. And unless Elizabeth tells us otherwise, I think we're done. And I want to wish everybody a happy rest of their day. Is that fair?
That's fair. Thank you so much to everybody. It was lovely to see you all, and I will close this out.
Take care.